Hello, and welcome. Tonight, we begin a journey through your own biology, through the invisible poetry of your body, and into the quiet future that's already unfolding inside your cells. You don't need to understand everything. You don't need to stay awake. You only need to listen and let the words carry you gently into rest. This is not a lecture. It is a story written in your blood, in your breath, in the spiral staircase of your DNA. We'll explore the science of CRISPR, a small, beautiful tool that can rewrite the human code, not through force, but with precision, not for power, but for healing, and perhaps even for design. Each chapter will take you deeper into the immune system, into the ethics of enhancement, into the future of health and human possibility, but always slowly, always softly, like drifting down a river of thought with the stars of your body twinkling above. Let your eyes close if they haven't already. Let your breath slow and your mind rest. You are safe here. You are held, and the story is just beginning. You lie still now, breathing gently into the pillow, your body warm with sleep's approach. Around you, the world fades. Walls melt into air. Noises blur into hush. But within you, a secret place begins to open. Deep beneath your skin, deeper than bone, past muscle and blood, and the quiet pulse of your heart, there is a garden you've never walked. This is your DNA, not a machine, but a living manuscript, written not with ink, but with letters curled in sugar and acid. It rests in every cell, trillions of times over, and yet it makes no sound. It does not ask for your attention. But tonight, we listen. You drift inward, as if shrinking gently, gently into one cell. A soft glow surrounds you. Everything here moves slowly. The world becomes small and humming. You are standing inside a quiet cathedral of life. The nucleus floats at the center, round and veiled, like a moon behind mist. Inside it, the strands lie coiled, long, ribboned spirals called chromosomes. They are tucked carefully, like threads in a loom, and if you looked closer still, you would see them unwind. You would see a double helix, two threads twining like ivy around a central dream. Each thread carries a code made of 4 letters, A, T, C, and G. They are not human letters, but molecular ones. Adenine, thymine, cytosine, guanine. But you don't need to memorize them. Tonight you simply feel them, tiny switches, dancing rhythms like notes in a lullaby, pairs with TC, pairs with G. Together, they form the blueprint of you. Every part of you, the curve of your hand, the color of your eye, the quiet way your heart responds to fear or music, began here. This garden shaped it. It is not perfect, not permanent, not fixed. It is a language always being spoken, always being read. Your body is a reader. Each cell takes from this scroll exactly what it needs. The recipe for a fingernail, a liver cell, a strand of hair. And then it carries out the instructions like a devoted artisan. No one teaches it how, it simply remembers. But the garden is not alone. Sometimes in a small moment of chance or inheritance, the letters may shift. One letter might change, a line may be skipped, a sentence repeated. These are mutations, tiny edits in the code. Most go unnoticed, some lead to change. This is how evolution has always walked, slowly, patiently, with quiet missteps that lead to wings or lungs or songs. As you float among the coils, you begin to see how vast this script truly is. If all your DNA were uncoiled and laid end to end, it would stretch to the moon and back many times. Yet it fits within a space smaller than a grain of salt, a library within a teardrop. But even now, in this gentle place, you sense the stir of something else. Not all visitors to this garden are kind. Far in the past, and still now, tiny intruders arrived, not through doors but through cracks in the code. Viruses, phages, invaders so small they had no life of their own. They couldn't breathe or grow without stealing your story. They came to write their names over yours. You don't remember them, but your cells do. Your immune system does. It has read the attacks and remembered the enemies. It has learned to guard the garden. This memory lives not in thought, but in chemistry, in the way a cell can recognize a stranger and call for help, in how it can cut out what doesn't belong. You don't think about it, but it's there, a sentry always awake, even as you sleep. And in the smallest of Earth's creatures, bacteria, something remarkable happened long ago. They too were attacked. They too were hunted by viruses. But they developed something different, a memory system written into their own DNA, a record of past invasions, a defense based on remembering. That memory is called CRISPR, and that is where the next part of your story begins. But for now, you rest beneath your ribs and skin in the quiet garden of your DNA. It is not silent here, it sings without sound. It sings of ancestors, of moments before you were born, of every cell that built the bridge between yesterday and you. You are made of instructions, yes, but also of adaptation, of patience, of countless edits that carried life forward. You are not only a reader of this book, you are its current page. Tonight, you lie here wrapped in the blueprint of yourself. You do not need to understand every code, every twist. It is enough to know it is there, keeping time, growing roots, opening petals deep within you. But something stirs near the edge of this garden. A whisper, not from your DNA, but from what tried to rewrite it. And if the garden can be read, can it also be rewritten? Can the thief's pen be turned into a healer's brush? You turn toward that thought, toward the old invaders and the new memory that emerged to face them. Toward Chapter 2, where the code remembers the breach, where we meet the quiet warriors who stand at the gate. You are still floating within yourself, drifting just beneath the surface of your thoughts. The garden of your DNA lies behind you like a dream half remembered, full of coiled beauty and quiet rhythm. But now the light has shifted. There is movement at the edge of the code, something once tried to enter. This chapter begins not in your body but far away, in a drop of water, warmed by sunlight, stirred by breath. In that drop, ancient and clear, live the first sentinels of memory. Bacteria. Bacteria are simple, single-celled creatures. They have no eyes, no limbs, no mouths. They are older than trees, older than bones. You could cradle 1 billion in your palm and never see them, and yet they have fought battles longer than any war you've read about. Their enemies are even smaller. Viruses known as bacteriophages or phages. Phages are not alive in the way you are. They are not built to survive on their own. They exist to invade. Like a key searching for a lock, they float until they find a cell, and then they latch on, injecting their own genetic code, a hijacking, swift and silent. Once inside, the virus uses the bacterium's own tools to make copies of itself. It turns the cell into a factory of infection, then bursts it open, sending new phages out into the world. One bacterium dies, then another, then thousands. For billions of years, this microscopic battle has continued. It is a war you were never taught, but it shapes the world. And then something changed. Bacteria, though simple, learned something profound: to remember. In the swirling oceans of early Earth, some bacteria began to keep pieces of the viral invaders. When a phage attacked, the bacterium snipped off a small fragment of the viral DNA and tucked it into its own genome. It filed the memory away in a special section, a kind of genetic scrapbook. This region was named CRISPR, clustered, regularly interspaced short palindromic repeats. Each piece of viral code was like a photograph of an enemy's face. The bacterium didn't just survive the attack, it kept a record of it, like a soldier sketching the shape of a sword that nearly struck them. The next time that same virus returned, the bacterium would recognize it. It would match the invader's code to the stored memory. Then it would call in an ancient guardian, a protein named Cas. Cas9, the most famous of these, acts like molecular scissors. Guided by the memory stored in the CRISPR sequence, Cas9 hunts down the matching viral DNA and cuts it. Precise, clean, no panic, no wasted motion, just removal. You picture it now, a library of dangers, a vigilant guard, a gentle snip in silence. This is immunity, not built on brute strength, but on remembrance. And it is not only bacteria that remember. Your own immune system holds a parallel truth. When a virus enters your body, say a cold or the flu, your white blood cells take note. They don't keep photographs, but they learn patterns. The shape of the virus, the way it behaves, the paths it takes. They study it like a spy learning the footsteps of an enemy. Then your body builds an answer. Antibodies, killer cells, messengers that call for backup. And once the danger passes, memory cells remain. Tiny sentinels that remember the face of the intruder just in case it comes back. You are alive because of this memory. You have walked through crowds and storms, touched doorknobs and railings, breathed air shared by strangers. And still your body remembers enough to keep you safe. But the memory held by CRISPR is something different. It is written into the DNA itself. It is passed on, not through experience alone, but through code. A mother bacterium that survives an attack passes its memory to its daughters. They are born ready, and one day human scientists noticed. They looked at the CRISPR sequences and saw something extraordinary. Here was a system honed by evolution that could recognize, remember, and cut DNA. Not only foreign DNA, but any sequence if guided properly. If that guide could be rewritten, if we could tell the scissors where to cut, then perhaps we could use CRISPR to edit genes. Not just in bacteria, but in plants, in animals, in humans. It would mean the power to correct a single letter in the vast novel of life, to stop a disease written into a child's blood, to silence a cancer before it speaks, to change what we once thought was unchangeable. But that is the story still unfolding. For now, you float back from the bacterial sea, back into the quiet of your own breath. Your cells are not battling at this moment, not that you feel, but inside, your immune system is still listening. The memory of past illnesses stirs faintly in the bone. Your body is not just flesh, it is story and defense and memory combined. You think of the bacteria again, floating alone, facing invaders, deciding not to forget. You think of CRISPR, not as a technology, but as an ancient instinct to protect, to learn, to adapt. And you wonder, if nature could invent such a precise system in silence, what might we do with it in full consciousness? The scissors wait. The memories wait. The code stands still, waiting for hands that might one day hold it gently, rewrite it carefully, like a poet correcting a line. But what does it mean to hold such a tool? And what happens when the scissors no longer just defend but begin to shape? You turn your thoughts now to the hands that discovered this system, to the moment Cas9 became more than a protector, to Chapter 3, where bacteria's invention becomes humanity's tool, where the scissors made of light prepare to cut. You drift now in the space between memory and discovery, carried softly from the bacteria's quiet defense into the hands of those who first saw its potential. The world outside your window sleeps, but deep in your mind, a new kind of light begins to flicker, the light of understanding. In a laboratory, far from any meadow or forest, scientists peer into drops of water, just like the ancient pools where CRISPR began. But they do not simply watch. They listen, not with ears, but with patterns, with microscopes, with intuition honed like fingertips brushing pages in the dark. They find the pattern, the repeats, the interruptions, the palindromes. They see the CRISPR system not as a curiosity, but as a codebook, one that doesn't just defend, it edits. And then, like unlocking an instrument string for the first time, they realize they can guide it. They can write the map. All it takes is a guide RNA, a short sequence of letters designed to match any place in a genome. Attach that guide to Cas9 and the scissors go where you tell them. They will find the matching sequence in the DNA, and they will cut. It is not a violent act. It is deliberate, measured, like trimming a single leaf from a tree, or removing a single word from a poem to make it sing more clearly. You imagine this process not with chemicals and pipettes, but with light. The scissors are not metal, but made of memory and precision. They float into the cell like whispers. They read the code, and if the target is found, they snip just there, only there, and then something beautiful happens. The cell, wise in its ancient way, sees the break. It does not panic, it repairs, it begins to stitch the torn thread. And if you give it the right thread to sew in, a correction, a small improvement, it accepts it, quietly, like it was always meant to be. This is gene editing, not just copying nature, but shaping it with reverence. And in 2012, two scientists, Jennifer Doudna and Emmanuel Charpentier, gave the world a gift wrapped in possibility. They published the first demonstration of programmable CRISPR-Cas9 editing. A tool once hidden in bacteria's survival became available to all of life. The scissors had moved from the cell's defense to humanity's imagination. You might think of a scalpel in a surgeon's hand or a brush in an artist's fingers, but this is more delicate than either. CRISPR moves inside the body, inside the cell, inside the nucleus, inside the spiral of DNA itself. It works where eyes cannot see. It whispers where voices cannot reach. And already those whispers are shaping change. In laboratories around the world, scientists begin to test. A faulty gene that causes blindness, corrected in mice. A mutation behind sickle cell disease, snipped and healed in human cells. Crops made more resilient to drought, without chemicals or guesswork, but by guiding the plant's own code to grow stronger. You feel a mixture now, calm and awe, comfort and curiosity. You are wrapped in the rhythm of it, recognition, precision, repair. It is not fast, it does not roar, it moves like a tide, steady, certain. But still, the question lingers in the silence. What else might we cut? The scissors do not know morality. They do not judge. They only follow the guide you give them. A guide to fix a broken gene, or one to change a trait before it ever forms. The same precision that can correct disease could be used to select for height, for eye color, for 100 preferences still undefined. That path lies ahead, just out of reach. But tonight is not for conclusions. Tonight is for feeling the shape of the tool. And so you return to your own cells, to the quiet way your body, right now, keeps time. There are letters moving, genes waking and sleeping, proteins folding like flowers, all without sound, all without struggle. If someone had told you as a child that you were made of code, you might have imagined wires, machines, screens. But now you know it is more like music, repeating themes, harmonies, soft corrections, crescendos hidden inside a breath. CRISPR is not a foreign force. It is built from something life already made. Its power is not in creating something new, but in guiding what is already there. You close your eyes and imagine light made into a blade. Not one that wounds, but one that prunes, that shapes, that tends the garden gently. And what would you choose if you could guide the scissors? Would you snip away the disease hiding in a strand? Would you cut fear from a memory or loneliness from a heart? Would you trim the sadness your child might inherit and replace it with calm? These questions drift like feathers. You do not need to answer. Not yet. For now, you rest in the knowledge that such a tool exists, that it works not through magic but through the logic of nature's own invention, that it was hidden in bacteria, waiting to be found, that it now glides through laboratories with hands steady and cautious, and tomorrow it may move through hospitals, through therapies, through the lives of those waiting for healing. The scissors are here, but what do we write now that we can erase? That answer waits in the next room of your mind, in a place where the code is no longer being trimmed, but rewritten, where we learn not just to cut, but to compose, where the letters of life become a language we speak back to the cell. And so you turn softly toward chapter 4, toward the moment the sentence begins again, not erased but rewritten with care. Welcome to the next page, writing in the language of cells. You are floating again, not in air or water, but in thought, drifting softly in the spaces between what is and what could be. Around you, the molecules of your body continue their silent dance. You are not asleep yet, but your breath has slowed. The night listens, and so does your body. Something ancient inside you is paying attention. Imagine for a moment that your body is a book, each cell a page, each gene a line, each letter a whisper passed down from your ancestors, written not in ink, but in the subtle geometry of molecules. You were written long before you were born. In that vast manuscript of your body, some pages are old, weathered by time, others are newer, barely touched, and some contain smudges, places where the writing faltered, a misspelled gene here, a repeated sentence there. These small slips, these inherited misprints, are sometimes harmless, but sometimes they echo as illness, disorder, pain. For so long we could only read this book. We studied it, we turned its pages, we wondered at its poetry, but we could not write back. Now that has changed. With CRISPR, we do not merely cut, we do not simply remove, we begin to compose. You imagine a quiet laboratory again, cool, clean, humming with low machines. At a bench sits a scientist, not with a scalpel or microscope, but with code, with a strand of guide RNA in one hand and a map of a human gene in the other. The target has been chosen, a single letter in the genome that spells pain or weakness or silence. But instead of deleting it, the scientist brings something new. A corrected letter, a restored word, a healthy sentence ready to take the place of the broken one. And the cell, clever and gentle, accepts it. It reads the corrected line and copies it forward. Through division, through time, through healing. You are watching gene expression become language. The DNA is no longer just a recipe. It is a story. And for the first time, we are co-authors. But this is not easy. The genome is a sacred text, shaped over billions of years. Every change must be made with care, with humility, with patience. Editing a gene is not like changing a tire. It's more like changing one note in a symphony and still expecting the music to play in tune. Every note you alter may ripple outward. A gene you fix in one place may speak to a dozen others. And so we learn to write slowly. In trials across the world, this careful writing is underway. In patients with genetic diseases like beta thalassemia or sickle cell anemia, scientists have used CRISPR to turn off a faulty gene and awaken another, older one, buried in the code. The results? Red blood cells that flow freely without the crushing pain that once defined life. In children with congenital blindness, gene editing has begun to write sight back into the eyes. In immune cells, CRISPR has helped train the body to fight cancers more precisely, more powerfully, and with fewer side effects. All of this has begun, not through miracles, but through understanding. But even now, as you lie still and calm, you wonder, can this language be learned by all? Can we teach the body to heal itself, not through drugs or surgery, but by writing the right instructions within? What if, one day, your body could edit its own mistakes? What if you could write away disease like crossing out a line in a poem and replacing it with a better one? That day may come. But for now, we begin with small edits, quiet changes, a single letter, a single child, a single life, improved. This writing, this gene editing, is not loud. It moves with the patience of moss growing across stone, and yet, within its quiet steps, the future takes form. Still, there are mysteries left in the code. So much of your DNA remains unread, not because it is silent, but because we do not yet understand its voice. Whole regions of your genome stretch like empty pages, filled with marks and patterns we cannot translate. Scientists once called it junk DNA, but now we suspect it may be the heart of something we do not yet know how to name. To write in the language of cells, we must first listen. We must become fluent not just in fixing, but in respecting, because to write is to take responsibility, to place your hand on the original manuscript of life and say, I understand this line, I will help it speak more clearly. It is an act of great hope and great humility. You now return in your imagination to your own body, to the quiet garden where we began. You think of your cells, your genes, your coded letters, millions of poems inside you still unfolding, still reciting. Your body is not a finished book, it is a living one, and now we hold the pen. But where do we go from here? Do we write only to heal, or do we write to prepare? To build immunity not just for one illness but for all? To give the body not just defense but design? To gift the next generation a sentence that begins, "You will not suffer what I did"? This is where our next chapter waits, where the story moves from repair to readiness, where the immune system becomes a page we can write on, not just to remember illness, but to imagine wellness, to not only heal the body, but to prepare it for what's to come. And so you drift forward now toward chapter 5, toward the secret shelves of your body's defenses, toward the place where white blood cells read rewritten lines, toward the immune system's secret library. You lie in stillness, your breath slow and soft, as if your body already knows it is time to listen, not to voices outside, not to stories on screens, but to the quiet archive that lives within you, an ancient library hidden in your bones, in your blood, in the rhythm of your immune system. This library does not look like shelves or scrolls. It is not built of books. It is built of memory. You are carrying it with you now, just beneath your skin, behind your eyes, in every lymph node and white blood cell. A place of records written not in paragraphs but in proteins. It is your immune system's secret library, and it remembers everything. When you were a child, the first time you touched a doorknob smeared with germs, your body did something astonishing. It didn't just react, it remembered. It built a profile of the virus that tried to enter. It crafted a response, cell by cell, and filed that lesson away. Each fever you survived, each cough you conquered, each vaccine you received, all stored in pages too small to see, pages that pulse quietly in your bloodstream. But this isn't just a library of the past, it's also a system of surveillance. Even as you rest, your immune cells are patrolling you, gentle, watchful, faithful. They move like moonlight through tissue, scanning for patterns they've seen before. They are detectives, scribes, warriors, and teachers, all at once. At the heart of this system are two remarkable tools, T cells and B cells. You do not need to remember their names. Just know this. Some of your immune cells carry weapons and some carry stories. The stories are antibodies, tiny Y-shaped molecules that fit a specific virus like a key fits a lock. Once your immune system creates the right key, it makes copies, it stores them, and those copies wait for years, sometimes decades, until the invader returns. And when it does, the response is faster, stronger, wiser. This is the idea that stirs now, healing without scars. You remember the wound that once marked your knee, or the ache that once curled inside your chest. Those memories are woven into you, not just as pain, but as resilience, as recovery. The body learns, the body adapts, but even healing leaves traces, fibers that grow back differently, proteins that tighten like old thread. And some wounds, especially those of the blood, the brain, the genes, never fully fade. CRISPR, though born in defense, offers something new, the possibility of healing that leaves no mark, correction so early, so precise, that the body never recognizes it was harmed. The story of the wound is never written. This begins before birth. In laboratories guided by caution and ethics, scientists are exploring the possibility of editing embryos, cells not yet formed into hands or hearts, but already holding the code for everything that will be. What if we could correct a life-altering mutation before the child is born? What if we could silence the gene that causes Huntington's disease, or change the base that brings on cystic fibrosis, or remove the seed of blindness before the eyes open? This would be healing in the deepest sense, not erasing what has been suffered, but preventing the suffering from ever blooming. And yet, this vision walks beside a shadow, because editing the unborn changes more than one life. It echoes forward. The edits pass on, like echoes into generations we will never meet. So we step with care. Ethicists, doctors, families, and governments speak in circles of conversation. How far should we go? Who decides what is a defect and what is simply difference? Will we write compassion or control into the code? Because it is tempting, isn't it? To remove not just disease, but discomfort. To choose not just wellness, but excellence. But healing without scars does not mean perfection. Perfection is brittle. It is static. It does not breathe. Healing without scars means grace. It means a kind of mercy coded into the cells. And it isn't limited to birth. Even in adults, healing without scars is beginning to unfold. In trials, scientists are using CRISPR to edit the cells inside the body directly. A person with sickle cell disease has stem cells removed, edited, and returned, now with instructions to produce healthy red blood cells. The painful episodes of the past begin to disappear, not with pills or transfusions, but because the underlying story has been rewritten. No stitches, no lasting mark. The wound never forms. In the retina, CRISPR is being tested to correct genetic blindness, delivering the editing tools into the eye itself, not with scalpels but with viral vectors, gentle messengers that carry instructions like whispered recipes. If it works, the person may see again, without ever having lost their sense of self. And in the liver, in the blood, in the bone marrow, more trials continue. Each one asks the same question. Can we repair what was broken so perfectly that it leaves no trace? You float through these images like a quiet observer. You watch edited cells multiplying silently, gently integrating with the body's rhythm. The immune system does not reject them. The tissues do not resist. The change is accepted as if it had always been. And you wonder, what kind of future lies in this path? Perhaps a time when newborn screenings don't just diagnose but correct. When medicine isn't a response to pain but an invitation to wellness. When hospitals become places of prevention, not just repair. But again, you hear the caution. This power, this ability to shape the body from within, must be held like flame cupped in palms, with reverence, with restraint, because scars also teach. They remind us of what we've survived. They ground us in humility. And not all scars are harmful. Some are cultural, ancestral, emotional, lines drawn in the story of who we are. To remove all pain is not always to grow. Some difference is not dysfunction. Some variance is not error. CRISPR must not be used to edit away the soul. And so the promise of healing without scars is not about perfection. It is about timing, about acting early, about treating illness not after it has worn us down, but before it has taken root. A kind of editing that honors the body as it is, while gently lifting the weight it need not carry. You think now of your own life, the invisible bruises, the unseen repairs, the moments your body mended you, even when you weren't aware. What if you had been spared one of those pains? What would your life be now? Different, better, or simply lighter? You breathe slowly, not to answer, but to hold the question. because the real gift of this kind of healing is choice, the choice to act early, to be kind to the future, to offer the next generation less pain, more ease, deeper readiness, and perhaps to rewrite not only the genes of the body but the shape of medicine itself. You turn now toward the edge of this idea, beyond prevention, beyond repair, to a place where design becomes destiny, where CRISPR is not used just to fix or avoid, but to optimize, where the body is crafted with intention, not just for survival, but for strength. You lie still beneath the weight of the night, wrapped in breath and thought. The silence around you is not empty. It's filled with questions too quiet to ask aloud, the kind that slip into your heart when the world has hushed. You feel your pulse, slow and steady, and wonder, what if this body could be more? The chapters behind you have unfolded the gift of CRISPR, a quiet tool, born of nature, handed to science. You've seen it fix the broken, prevent the suffering, rewrite the inherited pain. But tonight you float towards something more complicated, not disease, not healing, enhancement. You feel the shape of the word as if holding something warm and bright. It flickers with hope, but it casts a shadow too. To heal is to restore, to enhance is to choose. And choice always carries the weight of intention. Already the possibilities whisper around the edges of reality. In mice, scientists have used gene editing to increase muscle mass. In animals, to boost endurance. In crops, to enhance nutrition. These are not distant futures. They are now. And though human enhancement remains cautious, the path is opening. What if CRISPR could make your body stronger, your brain faster, your aging slower? Would you say yes? Would you say yes for your children? You imagine a parent holding a newborn in their arms, fragile, perfect, full of unknowable futures. And somewhere in a lab, a list is unrolled. Genes for intelligence, genes for height, for memory, for risk tolerance, for metabolic speed. Which ones do you choose? Which ones do you change? Which ones do you leave untouched? And more importantly, who gets to choose? Because enhancement is not simply about biology. It is about access, justice, power. If only some can afford to enhance, what happens to the rest? Will the edited become a class apart, born not better in heart or soul, but in engineered advantage? It's not hard to imagine a world where enhancement becomes expectation, where resumes include edited traits, where schools sort by genome, where beauty becomes a blueprint and deviation is labeled defect. You feel the tension gather in your chest. This is not just science, it's philosophy, it's culture, it's morality written in molecules. And yet the potential still sings. CRISPR may help reduce suffering not just by preventing disease, but by minimizing natural disadvantages. Could we give every child the same robust immunity, the same chance at focus and learning? Could we one day erase the boundaries that separate those born with luck from those born without? Perhaps. But to pursue that dream, we must first agree on something deeper. What is the purpose of the human body? Is it to perform, to compete, to conform, or is it to express, to adapt, to reflect the wild variety of experience? The body is not just a machine. It is a story, a lineage, a landscape. And editing it is not like fixing a clock. It's more like rearranging a constellation. Every star matters. Every silence between them speaks. So where do we draw the line? Many ethicists suggest we distinguish between therapy and enhancement. Fix what is broken, they say, but do not sculpt what is already whole. But even that line blurs. If we remove the gene for deafness, are we healing, or erasing a culture built on sign and song? If we edit for taller stature, are we helping a child thrive, or feeding a bias? The science is precise. The consequences are not. And so we return to the truth. CRISPR is a mirror. It reflects not only our knowledge, but our values, not only our skill, but our hunger. To guide this mirror wisely, We must decide together who we want to be, not as individuals, but as a species. Some say we should stop, that editing the human germline, the inheritable code, is too dangerous, too irreversible, that once we open that door we cannot close it again. Others say we must go forward, that refusing to improve life where we can is its own kind of harm, that to not act when we have the tools is to accept preventable pain. There is no simple answer, but perhaps there is a compass: humility, a deep, patient listening to voices beyond the laboratory, to cultures who see difference not as defect, to parents who love children as they come, to those with disabilities who do not wish to be edited away but understood. Enhancement must never become erasure, and so you breathe slowly now, feeling the truth settle into you like mist. The question is not only what CRISPR allows, it's what we believe is worth doing, because genes are not just code, they are memory, they are meaning, they are possibility folded in protein and chance. To edit them is not simply to cut and paste, it is to inscribe a new version of the future, and the future must be written by more than scientists. It must be shaped by communities, by elders, by dreamers, by those who have known pain and still found joy. You lie here, in the quiet, with the vastness of it all, a single body, a single breath, and yet part of something so much larger. You are not just a carrier of genes, you are a guardian of them, and perhaps that is what we all must become now, and guardians, not gatekeepers of progress, but stewards of compassion, not rulers of the genome, but partners in its unfolding, not editors chasing perfection, but storytellers asking again and again, what kind of life do we wish to live? You turn now gently toward the next chapter. The light has grown sharper, the questions heavier, because with power comes responsibility, and with editing comes the need for protection, for oversight, for ethics, for collective care. We have held the scalpel, now we must hold the shield. You rest at the edge of something vast, not sleep, not thought, but that tender space where the future breathes quietly. Your breath slows. The questions of the last chapter still echo softly inside you. If we can edit the body, what will we become? If we can design the future, who will guard it? This is where we arrive now, not at the edge of science, but at the threshold of stewardship, because power, especially biological power, must be held with care. The hand that edits must be guided by more than knowledge. It must be guided by ethics. And so, across the world, there is a quiet movement rising, a chorus of caution, not to stop science, but to shape it wisely. They are the guardians of the code. You imagine them now, not as soldiers or watchers, but as listeners, bioethicists in conference halls asking slow questions, lawmakers sitting with geneticists, drafting frameworks that stretch into generations, community elders, religious thinkers, patient advocates, each holding a piece of the truth. They do not speak in absolutes, they speak in nuance. They understand that CRISPR does not come with a moral compass. We must build that ourselves. And so, the first principle whispered through these circles is simple. Just because we can does not mean we should. You recall the story of the Chinese scientist who, in 2018, edited the embryos of twin girls to make them resistant to HIV. It was hailed as the dawn of a new era and condemned as a reckless breach of trust. The global scientific community responded with swift disapproval, not because the goal was evil, but because the process was rushed. The children could not consent, the risks were unknown, the lines, ethical, medical, social, were not respected. This became a turning point, a call for reflection, a signal that gene editing must not be a solo experiment. It must be a global dialogue. And so, guidelines began to form: international summits on human genome editing, scientific moratoriums on inheritable edits, advisory councils that included not just scientists but the public. Because the body is not a laboratory, it is a legacy. You feel it now, how delicate the balance is. CRISPR is not dangerous because it cuts genes. It is dangerous if it is used without accountability, without shared understanding, without limits. And so, more guardians gather. There are regulatory bodies like the FDA, the WHO, the national academies, who review every trial, every proposed therapy. They ask, is this safe? Is this necessary? Is this fair? There are ethics review boards who weigh not just the benefits but the harms, who speak for the voiceless, the unborn, the marginalized, the future. And most importantly, there are communities, people like you, parents wondering what choices to make, patients weighing hope against risk, citizens learning what it means to live in a world where DNA can be rewritten. These guardians are not gatekeepers, they are stewards. They understand that science without ethics is not progress. It is peril. They ask questions no machine can answer. What counts as consent when edits affect generations? Who ensures that enhancements don't widen inequality? How do we protect genetic privacy in a world hungry for data? Because your genome is not just information, it is identity, it is intimacy, it is yours. And like any precious thing, it must be guarded. Already debates stir over gene patenting, whether companies can own the rights to parts of the human code. There are concerns about insurance discrimination, genetic surveillance, the commodification of traits. The tools are powerful, but so are the temptations. And so the guardians work slowly, deliberately, asking not only what CRISPR will do, but what kind of world it will leave behind. You imagine for a moment the future. A child sits in a classroom, learning not only history and math, but how their body was edited. Not because something was broken, but because a choice was made. Will they feel gratitude or confusion? Will they wonder what they might have been had the code been left untouched? You don't need to answer. You only need to hold the question. Because to guard the code is not to resist change, it is to guide it, to make sure that the edits we choose are done with consent, with justice, with compassion. And so you too become a kind of guardian. You are not a scientist, but you are a voice, you are a witness, you are part of the future that will shape this path. In your questions, in your choices, in your conversations with others, you help decide what CRISPR will mean, not just in the lab, but in life. You lie still now, but your body hums with knowing. Every cell within you holds code, fragile, beautiful, unfinished. It does not need to be perfect. It only needs to be honored, and that is the true role of a guardian, not to protect perfection, but to protect dignity, to protect diversity, to protect the wild, unpredictable, deeply human journey that every genome carries. You have traveled far from the first chapter, where the double helix spiraled in your mind like starlight. You have seen how CRISPR can cut, can heal, can even imagine. But now you see the truth beneath it all. The future is not written in genes alone. It is written in values, in vigilance, in care. And so you close this chapter with quiet resolve, knowing that there will always be a need for guardians, not of the science itself, but of the soul behind it. You breathe out, long and slow. You are not alone. And as you turn now toward chapter 9, the world begins to open again, not with fear, but with responsibility. Next comes the future of health itself. How will medicine evolve when genes are no longer fate? What happens when we no longer wait for illness, but design well-being into our bodies? You breathe in slowly, the quiet around you wrapping like a blanket woven from memory and breath. The journey has taken you deep, through the spiral of your DNA, across the mysteries of immunity, into the hopes and cautions of human design. Now, gently, you arrive at a new threshold. The body no longer feels like a mystery you must solve. It feels like a space you are beginning to understand, a home under constant renovation. And in this new age, medicine is not just a response to crisis, it is becoming architecture, the architecture of health. You imagine your body now as a carefully designed structure, walls of muscle, corridors of blood, a roof of skin, a foundation built from bone and time. But unlike a house that erodes with years, this body, your body, may soon be sustained, refreshed, even enhanced from within. The question is no longer just, how do we treat disease? It is, how do we build lifelong health from the cellular ground up? Already, science is shifting. Where once medicine waited for pain to appear, now it whispers to risk before it speaks. Where once doctors treated illness, now they trace the roots of wellness. This is where CRISPR returns, not as a cure, but as a cornerstone. You see it now used not only to fix, but to predict. Imagine your genome as a blueprint. In it, quiet risks slumber. The faint outline of a cancer that might bloom in your 50s, a rare condition hiding behind your cholesterol, a tendency toward inflammation written like fine print. With gene sequencing, these risks can be read. With CRISPR, they can be redesigned. You may never know the disease that never had the chance to arrive. You might live into old age never feeling the quiet storm you were born carrying because the winds were calmed before they gathered. This is not immortality. It is a reimagining of maintenance, of long-term cellular care. Medicine, once reactive, becomes proactive. Doctors become architects, building strength, not just stitching breaks. You see this already, quietly growing. Gene therapies for rare diseases that once ended childhood in grief now offer long, healthy life. Inherited blindness, spinal muscular atrophy, even some forms of sickle cell anemia, they are no longer endings. They are beginnings of another way. And beyond rare conditions, we step into the everyday. Imagine heart disease, the world's quiet killer, dramatically reduced because the genes that influence cholesterol production have been peacefully edited. Imagine autoimmune conditions softened because the immune system was nudged gently toward tolerance before it ever turned on itself. Imagine Alzheimer's delayed or prevented. Not because of a miracle drug, but because early in life, the genetic architecture of neural resilience was gently strengthened. The body becomes a garden, medicine becomes the soil, CRISPR becomes the gardener's hand, patient, careful, attentive. But architecture is not only function, it is also form. As we move toward preventive care and genetic fine-tuning, we must ask, what does it mean to be healthy? Is it the absence of pain, the presence of potential, the ability to adapt, to recover, to grow? In the architecture of health, these answers matter, because if health becomes design, it must still honor the body as it is, not as a machine to be perfected, but as a landscape to be nurtured. There is room here for both technology and tenderness, for both editing and acceptance. You feel it now in your own breath. The desire not only to live longer, but to live more gently, to age with energy, to rise each day with the inner scaffolding quietly supporting you. This future may look less like hospitals and more like home-based monitoring, tiny sensors that read your blood, your breath, your genome, and speak to you before imbalance becomes illness. It may look like personalized diets crafted from your microbiome, designed to soothe your specific gut, your unique immune rhythm. It may look like vaccines not made from weakened viruses, but from gene sequences tailored precisely to your risk profile. It may even look like stem cells edited with CRISPR, reintroduced into your organs not when they are damaged, but before. You don't simply wait for the house to leak, you maintain it in peace. And yet, within all this, the human part of you stirs. Will this future belong to everyone, or will health itself become another form of inequality? Because a well-designed home means little if others live outside its walls. So as we build this architecture of health, we must build it wide, inclusive, equitable. No one should have to choose between health and poverty. No child should be left behind because their genome was never read or their mutation was never corrected. The future must not be a gated clinic. It must be a shared garden. And so we return, once again, to care. Care not only as treatment, but as structure. Care that begins before birth. Care that continues quietly, even when the body feels fine. Care that listens to genetics, but honors experience. Care that understands that health is not only in genes, but in joy, in safety, in rest. You lie here now, your own structure strong and soft, your breath like a foundation stone. The body you carry is not perfect. It never needed to be, but it can be supported. Its risks can be rewritten. Its rhythms can be respected. Its future can be protected gently from the inside out. You feel gratitude now, not for what has been fixed, but for what may never break, not for miracles, but for foresight. And as your breath deepens into rest, you hear the next question rise. If we can build health into the body, what else might we build into the human experience? If CRISPR can write wellness, can it also write resilience? Can it write adaptability? Can it help us evolve not just as individuals, but as a species, into a future shaped not by accident, but by quiet, ethical intention? And so you drift gently into chapter 10, toward the final unfolding, not the end. The night deepens around you, vast and weightless, like a sea without shore. You are afloat in thought now, your breath slow and full. This chapter does not rise with urgency. It unfolds like morning light, soft and gradual. You have walked through immunity, through memory, through healing and ethics and architecture. And now, the final question hums softly beneath your ribs. What kind of human might we become? Not by chance, but by choice. This is the edge of the story, where biology meets intention and evolution takes its next step not on its own, but with us beside it. For millions of years, evolution has sculpted life with blind hands. Natural selection, genetic drift, environmental pressure. The rhythm was slow, shaped by accident, filtered by survival. You are here because of it. Every part of you a result of what once helped your ancestors endure. But now, with CRISPR in hand, you begin to turn the chisel toward yourself, no longer waiting for nature to choose. Now we ask, what do we want to become? This is not the start of something unnatural. It is simply a shift in authorship. Because CRISPR does not erase evolution, it joins it. It makes it conscious. You imagine your genome as a manuscript written across generations, paragraphs shaped by famine, by fertility, by climate and migration. But now, with this quiet tool, we are scribbling margin notes, adjusting grammar, rewriting chapters. The question is, how do we write with wisdom? Already, small edits are suggesting big changes. Genes that influence muscle mass, genes that affect memory, genes linked to emotional sensitivity, social behavior, even sleep. If we learn to adjust these gently, predictably, inheritable across generations, we are no longer treating illness. We are redesigning the species. You lie still now, feeling the gravity of that. It is not frightening, but it is sacred, because to design a future body is also to design a future mind, a future culture, a future soul, and not all edits are physical. What if we learned how to reduce the genes that influence violence, or enhance those that promote empathy? What if we could encourage neural plasticity, so that each human mind becomes more adaptable, more open, more resilient? What if evolution is not simply about becoming stronger, but becoming kinder, more cooperative, more attuned to the fragile ecosystems we live within? Because humans do not live alone. We are woven into forests, oceans, soil, sky. If we evolve by design, we must do so not just for ourselves, but for the world that holds us. The bee that pollinates your food, the bacteria that live in your gut, the coral that shields the coastline. the ancient seeds still sleeping in Arctic soil. You are not separate from this web, so your evolution, if guided wisely, must not dominate. It must harmonize. Perhaps the future human is not taller, faster, more intelligent in a narrow sense. Perhaps they are gentler, more responsive, more attuned, able to shift genetically in response to a warming climate or a new virus or a changing planet. You lie here and imagine it. A child born with genes designed not for competition, but for cooperation. A generation of humans whose bodies are designed not only to survive, but to restore, to regenerate tissues, to resist panic, to learn languages easily, to laugh often, to sleep deeply. This is not utopia, it is potential, but it will not arrive by accident. It must be designed with ethics, with humility, with foresight stretching into centuries, because genes do not stop with one person. They ripple forward, every edit echoes, and so the evolution we choose must not be rushed. We must protect what makes us human as we shape what humanity may become. You may wonder, will there still be failure in such a future? Will there still be sorrow? Will the human experience lose its grit, its friction, its beauty? But listen closely. Evolution does not promise peace. It promises possibility. And designing it does not remove hardship. It simply gives us the chance to shape how we respond, how we grow through it, how we carry one another through. You are part of that design already. Every time you ask what kind of world we're creating, every time you listen to science with a heart open wide, every time you choose compassion, even in the smallest way, you are shaping evolution, not in a petri dish, but in the quiet space of culture, of family, of care. Because our bodies are one layer of change, but our choices, our values, are the deeper ones, and they too can be inherited. You drift now into the softest space of thought, not a destinations, but a becoming. You see your future descendants, walking calmly through forests that breathe cleaner air, children growing without the burden of inherited pain, elders whose memories stay intact through time, communities that do not fear illness but understand it, not perfection, not control, but harmony, a human species learning, again, how to live within itself and within the world. The final breath of this chapter is not a conclusion. It is a seed planted gently in your imagination. Let it grow, let it guide, because evolution by design is not about power. It is about responsibility, and that begins now, with you, in your body, in your breath, in the choices you make each day. Let your life be a part of that story. Let your genome carry not only code, but kindness, not only strength, but wonder, not only data, but dreams.